Hello everyone, my name is Tamika and today we're going to discuss the main steps in the process of DNA replication. First we'll do a brief overview and then we'll describe the steps individually. When a cell divides, each daughter cell must receive its full complement of genetic material in the form of chromosomes containing DNA. A chromosome is made up of two long strands of DNA and several types of proteins. A DNA strand is composed of a long backbone of sugar and phosphate units. One of our different nucleotide bases, A, T, C, or G, hang off each sugar unit. The sequence of the bases encodes genetic information. The three steps in the process of DNA replication are initiation, elongation, and termination. Replication depends on the pairing of bases between the two strands of DNA. The A base can only bind to a T and the C can only bind to a G. In the DNA double helix, the bases of one strand face across and bind to those of the other strand. Therefore, the base sequence of each strand complements that of the other. The sequences are anti-parallel and serve as templates for each other's replication. The two strands are labeled by the location of certain chemical bonds in the DNA backbone. The cell can replicate the leading strand as a single unit, but must replicate the lagging strand in small pieces. Replication begins at a location on the double helix known as ORIC, to which certain initiator proteins bind and trigger unwinding. Enzymes known as helicases unwind the double helix by breaking the hydrogen bonds between complementary base pairs, while other proteins keep the single strands from rejoining. The topoisomerase proteins surround the unzipping strands and relax the twisting that might damage the unwinding DNA. The cell prepares for the next step, elongation, by creating short sequences of RNA called primers that provide a starting point of elongation. With the primer as the starting point for the leading strand, a new DNA strand grows one base at a time. The existing strand is a template for the new strand. For example, if the next base on the existing strand is an A, the new strand receives a T. The enzyme DNA polymerase controls elongation, which can only occur in the leading direction. The lagging strand unwinds in small sections that DNA polymerase replicates in the leading direction. The resulting small Okazaki fragments contain 1,000 to 2,000 bases in bacteria, but eukaryotes have fragments of only 100 to 200 bases. The fragments terminate in an RNA primer that is subsequently removed so that enzymes can stitch the fragments into an elongating strand. After elongation is complete, two new double helices have replaced the original helix. During termination, the last primer sequence must be removed from the end of the lagging strand. This last portion of the lagging strand is the telomere section, containing a repeating non-coding sequence of bases. Enzymes snip off a telomere at the end of each replication, leading to a shorter strand after each cycle. Finally, enzymes called nucleases proofread the new double helix structures and remove mispaired bases. DNA polymerase then fills in the gaps created by the excised bases. Now, to describe the steps individually. First, we have the parent molecule. It has two complementary strands of DNA. Each base is paired by hydrogen bonding with its specific partner, A with T and G with C. The first step in replication is separation of the two DNA strands. Each parental strand now serves as a template that determines the order of nucleotides along a new complementary strand. Then the nucleotides are connected to form the sugar-phosphate backbones of the new strands. Each daughter DNA molecule consists of one parental strand and one new strand. And that pretty much sums up the process of DNA replication. Hopefully, this was able to help someone. Again, my name is Tamika. Thank you all for joining me today. Information provided by SeattlePI.com with illustrations provided by Pearson Education. Thank you.